Hey guys, it's Callie, and I'm going to be talking to you about INK4. INK4 refers to a family of cyclin-dependent kinase inhibitors, including P16, P15, P18, and P19, and they're part of a family that is kinase-specific. They inhibit the cyclin-dependent kinase CDK4, as well as CDK6, but they get their name after CDK4. The normal function of these proteins is to block progression of the cell cycle beyond the G1 restriction checkpoint. The G1S checkpoint in the cell cycle occurs at the end of the G1 phase before S phase, so before DNA synthesis and replication. This checkpoint becomes a stopping point for cells that have DNA damage or are contact inhibited, meaning they need other cells to touch them to provide cues to grow and divide themselves. Normally, RB binds E2F transcription factor proteins when it is hypophosphorylated and active, which inhibits cyclin transcription, as well as other key genes needed to progress through the cell cycle. In response to environmental signals, the INK CDKAs will complex with CDK4s and 6s and prevent them from binding to cyclin D and phosphorylating RB, which promotes entry into S phase, or RB does. In other words, their function is critical to preventing cell cycle progression because they promote hypophosphorylation of RB or prevent its phosphorylation. When RB isn't phosphorylated or is hypophosphorylated, its conformation is such that it can bind to E2F and prevent gene transcription necessary for cell cycle progression. When it's phosphorylated, it can't do that. Now, which INK4 family member inhibits CDK4/6 activity appears tissue-specific. P18 and 19 oscillate throughout the cell cycle, but levels of 15 and 16 remain constant. For example, INK4 family members all act in the pathway to prevent cyclin-dependent kinases from allowing cell cycle progression from G1 to S phase, but their presence, whether cyclical or consistent, and their stability are tissue-specific, which is what accounts for the various different neoplasms these gene mutations are involved in, which I'll talk about. But 15 and 16 are constant throughout the cell cycle, but are acted upon by TGF-beta to prevent cyclin D binding to CDKA4 and 6. TGF-beta binds to its receptors and activates its intracellular pathway that involves binding of SMAD3 and 4, which activates cyclin-dependent kinase inhibitors P15 and 16. For P18 and 19, this response is induced by mutagens instead of TGF-beta, but in either case, this family of proteins binds and inhibits CDK4 and 6 activity. Obviously, mutation events in these proteins can be carcinogenic, particularly when they accumulate. There's nothing to stop CDK4 and 6 binding to cyclin D and promoting RB phosphorylation and dissociation from E2F, thus facilitating constitutive progression from G1 to S. Or there's nothing to stop them when they've accumulated mutations. Many cancers associated with mutations in members of the INK4 family, for example, P16 mutations are linked to squamous cell carcinomas in the areas of the head, such as the mouth and lips and gums, as well as cancers in the lung, pancreas, brain, and bladder, just to name a few of many. These can be adenomas or carcinomas, among others. P19 mutations are associated with similar cancer locations and types, as well as unique ones. Some include breast and uterine and ovarian cancers. These types of cancers, in this case, include adenomas, adenocarcinomas, and again, others. Epithelial cancers. A significant portion of P18 mutations are found in uterine neoplasms as well, even more so than P15. Cancers of the hematopoietic and reticular endothelial systems are more common in P15 mutations than in other INK4 family members. P19 mutations are associated most commonly with carcinomas and adenocarcinomas of the stomach. It's unsurprising that there are many polymorphisms in these genes associated with cancer, and the consequences of their mutations vary. For P15 or P16, for example, consequences of substitution mutations can include splicing mishaps with donors and acceptors, premature stops, missense mutations, frame shifts, and more. They result from, and these mechanisms and results are found in and associated with polymorphisms in other INK proteins as well. So P15, P18, P19. If you use the Cancer Genome Atlas, you'll see this displayed very clearly. Changes to protein coding sequences are a common method by which this gene becomes an oncogene, and in addition to both copy number gains and losses. In addition to these mechanisms, hypermethylation of P18, 15, 16, and 19 is seen in a variety of cancers and is also tissue-specific. In some cases, there are multiple causes, such as both copy number variant gains and losses resulting from mutations, which may occur in promoter regions of the gene. Changes to protein coding regions appear to be most common and occur when mutation, most often a substitution, renders the protein or product ineffective by changing the amino acid sequence or causing a premature stop or frame shift, depending on the localization of the mutation. Fun fact, fish can get melanoma. There are groups of fish located along the equator called Zophophorus fish, which contain many copy number variants of INK4 family members associated with melanoma. You can see that here. Do you see, if you can see figure, it's all the way to the right. This fish clearly is hyperpigmented, and this is melanoma. The number of copy number variants indicates that INK4 has had a tumor suppressor activity or function for at least 350 million years. These are my sources. Thank you so much.